Welcome to our video about the fascinating world of Pokemon and their placement on the Tree of Life. Today, we take a closer look at Diglett and its relationship with all Loriciotheria species. Ready? Let's go! Moles are a family of mammals from the order Insectivora, Eulipotifla. They comprise more than 60 species spread across Eurasia and North America, with the European mole being the only representative living in Central Europe. The animals exhibit a wide range of adaptations to different lifestyles. Consequently, their external appearance is also highly variable. Generally characteristic are a cylindrical to spindle-shaped body with a short neck, topped by a pointed head. Most species, such as Eurasian moles and New World moles, live by burrowing underground. Their forelimbs are robust and shovel-like, and positioned far forward with palms facing outward. Additionally, water-dwelling forms like desmans and the star-nosed mole are included. The long hind legs of desmans are webbed. The above-ground shrew-like moles and different species of golden moles, on the other hand, resemble shrews in appearance. All moles have dense fur with predominantly dark colors. Except for the shrew-like moles, most representatives lack externally visible ears, and their eyes are often underdeveloped. A unique feature is the presence of Eimer's organs, which serve as sensitive tactile tools. Predominantly utilized habitats include open landscapes and river valleys, but also high mountain regions. Often, the presence of a thick, humus layer is a prerequisite. The behavior of only a few species has been studied in detail. Both the burrowing and swimming lifestyles required specific physical adaptations. The burrowing moles create complex tunnels and passages, whose surface characteristic is the molehill. The tunnels can encompass multiple levels and contain nest chambers. Near-surface passages are usually used for foraging. In water-dwelling species, foraging occurs in standing and flowing waters. The animals retreat to partially self-dug underground burrows for rest. This also applies to golden moles, whereas little information is available about shrew-like moles. Generally, moles are active both day and night. With few exceptions, they live solitary lives, with each individual claiming its own territory. The animals primarily feed on invertebrates. Mating usually takes place once a year. After a gestation period of about 1 month, 2 to 6 young are born. The offspring reach sexual maturity by the following year at the latest. Moles are generally rather small animals. They reach a head body length of 6.3 to 24.0 centimeters and a weight of 6 to 440 g. The smallest representatives include the ground-dwelling shrew moles, Eurotrichini, and the American shrew mole, Neurotrichini, while the Russian desman, Desmana, from the group of semi-aquatic adapted desmans, Desmanini, represents the largest form. The burrowing moles as the core group of the family, especially the true moles, Talpidae, and the New World moles, Scalopini, are between 8 and 23 centimeters long and weigh between 20 and 220 g. The tail length in moles is highly variable. In the burrowing species it is relatively short, often covered with only a few hairs that partly function as tactile hairs and allow orientation backward. In the shrew-like species and desmans, the tail is sometimes as long as the rest of the body. The latter have a partially or fully flattened tail, which is used as a rudder in the water. The body of moles is spindle to cylindrical shaped, with the head sitting on a very short neck. The fur covering the entire body except the feet and the tip of the nose consists of 2 hair layers in the underground living species, the wool hair and the outer hair. In the desmans, guide hairs are also present. The wool hair is fine and wavy in all moles. The outer hair generally consists of coarser hairs, which can be bristle-like in desmans. In the latter, the outer hairs are usually flattened, preventing air from escaping underwater. Overall, the fur is very dense. In some New World moles, it consists of up to 3,000 hairs per square centimeter, while in desmans, this increases to up to 20,300. The fur color varies from light gray through various shades of brown to black. A silky sheen often appears. The ventral side is only slightly lighter than the dorsal side. All species have skin glands that primarily serve to secrete sweat. Up to 600 such glands may cover a square centimeter of skin. The most noticeable are the musk glands at the base of the tail in desmans. The head appears pointed due to the characteristically elongated nose. The eyes of moles are very small and partly covered by skin, and ear pinnae are absent in all species except for shrew-like moles. The limbs of moles appear short and end in 5 clawed fingers or toes. The forelimbs of the underground living species are modified into digging tools. The hands, turned outward, are widened shovel-like. The limbs of desmans, on the other hand, have webbed toes and bristle-like hairs on the edges. They are only somewhat suitable for digging, although the former can partially dig their own burrows. The skull of moles is relatively uniform. It has a typically elongated shape with a broad braincase and a narrow rostrum, giving it a wedge-like appearance. The length-width ratio is about 2 to 1, but significant species-specific variations occur in the width of the rostrum. An obvious constriction behind the orbit, except in desmans, does not occur. In lateral view, the braincase only slightly rises from the snout region. Distinct bony ridges like a sagittal crest or an occipital crest are absent, with desmans being an exception. The individual skull bones fuse relatively early in individual development. The zygomatic arch is developed in all moles, but is rather weak. It does not protrude far, instead running parallel to the skull side. On the underside of the skull, the tympanic bulla is either fused with the skull as in Eurasian moles, Talpidae, Western American moles, Scaponus, the Eastern American mole, Scalopus, or the white-tailed mole, Parascaptor, or connected to it by connective tissue as in the American shrew mole or the star-nosed mole. Typically, the tympanic bullae are small and rounded. The auditory ossicles are not significantly enlarged in most species, while they are exceptionally small in the latter two forms. Only in the white-tailed mole and the short-faced mole, Scaptochirus, does the malleus have a distinctly swollen head. Both the connection of the tympanic bulla with the skull base and the development of the auditory ossicles reflect the stronger or weaker adaptations to underground life, which in turn affects hearing ability. Thus, animals living largely above ground perceive higher frequency sounds more, while those living underground perceive lower frequency sounds more. The lower jaw in all moles is slenderly built. The coronoid process is often rounded, the condylar process slender, and the angular process robust. The connection with the skull is hinge-like, allowing only upward and downward movements. Some species mainly from the group of New World moles and true moles, including the European mole, have retained the original number of teeth of higher mammals with 44 teeth. The most significant differences are found in the front teeth. This not only concerns the varying number of teeth, but also the formation of the teeth. For example, in true moles and the long-tailed mole, Scaptonechini, the upper canine is greatly enlarged, while all other representatives, except for the shrew-like moles, have a hypertrophied first incisor. In the latter, all front teeth are about the same size. The back teeth with the premolars and molars resemble those of other insectivores. The chewing surfaces are equipped with pointed cusps and sharp enamel ridges, which are adaptations to a carnivorous and insectivorous lifestyle. On the molars, the main cusps are arranged in a dilambodont, W-shaped, pattern. Notable is a strong oligodontia in some forms, especially the Eurasian moles and the East Asian moles, Majura, within the true moles. This can often be observed in the front teeth and premolars, and involves both missing and extra teeth. Another noteworthy feature in true moles, desmans, and the star-nosed mole, Condylurini, is the transition from milk teeth to permanent teeth occurring before birth. Shrew-like moles, New World moles, and the Japanese shrew mole, Urotrichini, on the other hand, exhibit a postnatal tooth replacement. The spine consists of 7 cervical, 13 to 14 thoracic, 5 to 7 lumbar, 5 to 6 sacral, and 8 to 27 caudal vertebrae. The number of vertebrae before the tail thus varies between 31 and 33, with the latter predominating. The spinous processes of the cervical and thoracic vertebrae are generally weakly developed. The thoracic vertebrae bear slender ribs. If a 14th pair of ribs is developed, it is significantly reduced and has no function. As highly specialized animals, moles have some peculiarities in skeletal structure, especially regarding the arms and shoulder girdle. The changes are more noticeable the more the individual species have adapted to a burrowing lifestyle. In general, the bones of the arms are more massive and broader in burrowing moles than in terrestrial or semi-aquatic forms. The shoulder girdle is usually shifted forward and lies above the last cervical vertebra in burrowing species, with exceptions in desmans and shrew-like moles. Unique among mammals is the articulated connection between the clavicle and the humerus, specifically with the greater tubercle of the latter. The usual articulation between the humerus and scapula is only present in shrew-like moles. The clavicle also plays a special role in the positioning of the arms. In ground-dwelling shrew moles, it is still long and slender, with some shortening compared to shrews. Nevertheless, the forelimbs are positioned under the body, allowing terrestrial locomotion. From the water-dwelling desmans to the burrowing forms, the clavicle progressively shortens until it reaches an almost cubic shape in Eurasian and New World moles. This direct articulation with the humerus allows the arms of burrowing moles to lie far in front of the chest next to the head, integrated into the body contour, facilitating digging movements underground. The scapula is similarly modified, becoming slenderer from ground-dwelling to burrowing representatives and losing prominent processes. In burrowing moles, the acromion regresses, and the metacromion, a process of the acromion, is reduced. The humerus undergoes significant changes, being relatively slender and round in cross-section in shrew-like moles and desmans, but short and robust in burrowing moles, making it the most massive bone in the entire skeleton. This is achieved by widening the shaft, flattening the bone, and extreme lateral expansion of the joint ends. The width-to-length ratio of the humerus in New World and Eurasian moles is about 0.33 to 0.45, while in shrew-like moles and desmans, it is 0.17 to 0.22. The short and broad humerus of burrowing moles serves as an attachment point for strong arm muscles that enable underground locomotion. However, the broad joint ends, primarily those of the elbow joint, prevent rotations in this area, allowing only up and down movements. Arm rotations occur from the shoulder, rotating the humerus along its longitudinal axis. Despite its massiveness, the humerus of burrowing moles lacks special bone condensing. The long, extended upper joint process of the ulna, olecranon, part of the elbow joint in burrowing moles, serves as an anchor for the arm muscles. In more terrestrial forms, this process is significantly shorter. Similar to the arm bones, the hand bones in burrowing moles are also greatly thickened compared to other representatives. Additionally, there is a crescent-shaped sesamoid bone, the so-called prepollex, os falciform. It attaches laterally to the scaphoid. In Eurasian and New World moles, it reaches the length of the carpal plus metacarpal bone and is rudimentary in shrew moles and desmans, but completely absent in shrew-like moles. The os falciform widens the palm as a sixth finger and supports digging underground. It is already formed embryonically. Unlike their arms, the legs of most moles are less specialized. The femur and tibia are usually the same length. Desmans are an exception, as the former is only half as long as the latter. The tibia connects with the fibula at the midshaft. The individual foot bones are long and slender. Some representatives, such as Eurasian moles, have a small sesamoid bone on the foot corresponding to the pretoe, prehallux. Its function is unclear, but it could be genetically linked to the formation of the prepollex. Few details have been provided about the internal organs of moles. The lungs are generally larger than in most mammals. For Eurasian moles, their share of body weight is nearly 1/5, while for desmans it is 1/10. By comparison, they account for less than 1/10 in shrews. The digestive tract shows a rather simple structure, like all insectivores. This also applies to the stomach, which in its pyloric extension resembles that of shrews but shows similarities to that of hedgehogs due to a conspicuous constriction between the pylorus and the stomach body. Compared to hedgehogs, the inner surface is enlarged, indicated by a higher number of glands in the stomach fundus. The distribution of glands forming the stomach lining is very characteristic of moles. About 83 to 89% are formed by fundus glands, 4 to 6% by pyloric glands, and 1 to 3% by cardiac glands, with the rest in transitional areas. In shrews, the proportion of fundus glands is lower. The stomach accounts for 15 to 25% of the head body length, being shortest in the American shrew mole, intermediate in burrowing moles, and longest in the star-nosed mole. The intestine forms a simple tube. Compared to body length, it is very short, and the cecum is absent. The snout region of moles is rich in sensory cells, called Eimer's organs. Named after Theodore Eimer, these are hump-like protrusions, with a diameter of 40 to 120 micrometers. The protrusions consist of invaginated skin marking the ends of nerve pathways, either free nerve endings, or those from Merkel cells. This allows the animals to likely perceive not only tactile stimuli but also electrical stimuli, enabling them to sense weak electric fields created by the muscle movement of their prey. This sensory system is most developed in the star-nosed mole, which has 22 finger-like projections arranged radially around the nose. These projections have an estimated 26,000 individual receptors. In other moles, the Eimer's organs are less complex or differently structured. They lack the finger-like tentacles of the star-nosed mole, so the humps are directly arranged on the nasal shield, which is divided into several parcels. There can be between 1,300 and over 2,400 individual receptors. In some forms, such as the short-faced mole or the Eastern American mole, the receptors may be reduced. In desmans, fine vibrissae sprout between the sometimes noticeably reduced humps. Only shrew-like moles show no evidence of Eimer's organs. The diploid chromosome set of moles consists of 30 to 48 chromosome pairs. In a larger portion of species, it is 2n equals 34, which is considered the original form. This version is found in the more primitive shrew-like moles, but also in some golden moles, the star-nosed mole, and the digging-adapted New World moles. It also appears in the similarly underground-dwelling true moles, where it underwent stronger modifications. Extreme values have been determined in this group. The simplest chromosome set is found in East Asian moles, while the most extensive is in the short-faced mole. Strong deviations from the original type are found in desmans and the American shrew mole. The assumption of a largely conservative development of moles regarding their cytogenetic properties cannot be confirmed by increasing analyses since the 2000s. However, it is still unclear why New World moles exhibit pronounced stability in their chromosome set while true moles do not. The fundamental number, the number of arms of autosome pairs, also varies significantly, ranging between 46 and 72. A bimodal distribution is notable, with one focus around 54 and the other around 62 to 64. Mutations causing single-armed autosomes to be converted into two-armed ones are likely causes. Almost all family members possess a large metacentric X chromosome and a small spotty Y chromosome. The mitochondrial genome of moles is relatively uniform among the species studied so far, with the number of base pairs ranging from 16,530 to 16,960. Moles are exclusively native to the northern hemisphere, namely Eurasia and North America. In Eurasia, they inhabit wide areas of western, central, eastern, and southern Europe, from the Iberian Peninsula, and occur from western Asia through central and eastern Asia to the Japanese archipelago. To the south, their range extends across mainland Southeast Asia to the Malay Peninsula. The North American distribution focus ranges from southern Canada to northern Mexico. Therefore, the animals inhabit both temperate zones and subtropical and tropical areas. Some forms are also adapted to boreal conditions. Moles are generally absent from polar and subpolar regions. Overall, the family members occupy diverse habitats. These range from open meadows and steppe areas to river valleys, coastal dunes, and semi-arid landscapes. Cultivated and garden areas are sometimes inhabited. Some species, like shrew-like moles, occur in extreme high-altitude regions up to 4,600 m above sea level. Moles usually prefer areas with moist soils and thick humus layers. In very dry regions or habitats with dry seasons, the animals are confined to areas with more humid ground or river meadows. In temperate regions, some species, like the European mole, the Eastern American mole, or the star-nosed mole, have extensive distribution areas. The occurrences of individual species are more limited in the rather semi-arid habitats of Western Asia and the southern Black Sea region, as well as in the tropical areas of Southeast Asia. These regions are considered significant diversification centers. In some parts of their distribution area, multiple mole species inhabit a region, such as the Mediterranean area or the Japanese islands, as well as in Southeast Asia. Species often differ in size and the use of specific ecological niches, so their co-occurrence is only partially syntopic. The partial morphological diversity of moles reflects their different adaptations to various habitats. Essentially, four ecomorphotypes can be distinguished: terrestrial, shrew-like moles; semi-aquatic, Desmond's, star-nosed mole; semi-fossorial, Japanese shrew mole, American shrew mole, long-tailed mole; fossorial, true moles, New World moles. The different ecological adaptations lead to varying lifestyles. Most representatives of moles live underground. They are adapted to this lifestyle with their cylindrical body, forward-shifted forelimbs, and outward-turned and broad hands that are transformed into digging tools. Since only the forelegs are used for digging, they are the most developed. The animals spend most of their lives in a self-dug underground tunnel system. The excavated material is often brought to the surface in the form of molehills. Depending on the species and habitat, these tunnels can extend just below the surface or into greater depths, comprising multiple levels. In cooler regions, moles often stay deeper underground in winter than during the warmer months. The tunnel systems contain several nest chambers padded with plant material and often positioned deeper in the soil. Sometimes very large, castle-like mounds are piled up. This is known from the European mole, but also from the Caucasian mole and the Townsend's mole. Most species can probably swim if necessary. However, semi-aquatic forms have emerged with Desmond's and the star-nosed mole. The animals spend most of their active time in the water and also search for part of their food at the bottom of bodies of water. The bodies of the animals are streamlined, and the forelimbs are not as characteristically forward-shifted as in burrowing moles. The star-nosed mole paddles with all four limbs in the water. In Desmond's, only the hind legs provide propulsion, which are therefore much more robustly developed than in burrowing moles. On land, the semi-aquatic moles use partly self-dug tunnel systems, sometimes also rock crevices, as retreats. The entrances are exclusively below the waterline, but the resting chambers are above it. Shrew-like moles are likely pure surface dwellers and probably search for their food there. Their body structure thus largely corresponds to that of shrews, with limbs under the body. The construction of the hands and feet allows digitigrade locomotion. However, almost nothing is known about their lifestyle. The same applies to the various golden moles. These can also run quadrupedally like shrew-like moles, but they also create simple tunnels and passages, which generally only shallowly extend into the ground or cross leaf litter layers. The tunnels are largely used for foraging but can also contain individual nest chambers. Life underground for the burrowing species required special modifications. Both the Eastern American mole and the European mole show adaptations to the low oxygen air in their tunnel systems in the biochemical properties of their hemoglobin. As a result, both species have an increased capacity for CO2 uptake, which in turn makes more oxygen available. The hemoglobin of the European mole has a higher oxygen affinity compared to exclusively surface-dwelling small mammals. Native P50 value equals 2.9 kilopascals compared to 6.5 kilopascals in mice, while the oxygen affinity of CO2-loaded carbaminohemoglobin is relatively high. The concentration of 2,3-BPG in the erythrocytes of the European mole is around 5.3 mol/L under normal conditions, which is not noticeably high compared to other mammals. Additionally, it varies between different mole species. The binding affinity of hemoglobin to 2,3-BPG is significantly reduced in some species, and is therefore discussed as another cause for the high oxygen affinity of hemoglobin, as well as its CO2 loading capacity. Due to the regression of the eyes, the sense of sight is greatly reduced and mostly limited to distinguishing between light and dark. In contrast, other sensory organs are sometimes extraordinarily developed. This includes the sense of touch, which plays an important role in the overall sensory perception. Part of this is taken over by the Eimer's organs in the nasal region, and additionally, numerous vibrissae are distributed over the snout and other body parts. Large areas of the neocortex of the cerebral cortex serve to detect and process tactile stimuli. The trigeminal nerve, which is complexly structured, acts as a messenger. In the Eastern American mole, this nerve has up to 30,000 afferents, or branches for signal transport, and in the star-nosed mole, there are up to 84,000. The extremely high value in the latter species is due to the finger-like skin appendages of the nose with the complex Eimer's organ system. This highly sensitive tactile organ enables the star-nosed mole to efficiently recognize, locate, and capture its mostly small prey. The resulting reaction times are among the fastest in the animal kingdom. In addition, the sense of smell plays an important role. For burrowing moles, it is crucial for orientation in the dark tunnels and locating prey. Experiments have shown that the Eastern American mole can perceive smell separately with both nostrils. This effect, known as stereo-smelling, increases the accuracy of localization. The semi-aquatic desmans and the star-nosed mole have developed techniques to capture scent traces underwater. This is done by expelling air bubbles, to which scent particles adhere, and then inhaling the bubbles. The social structure of moles has only been studied in a few species. Like most insectivores, the animals are generally territorial loners and react aggressively to conspecifics. Male and female individuals only come together during the breeding season. Individual territories can overlap between sexes, but not with same-sex individuals. Territorial markings are made with secretions from the anal glands. Only a few species share tunnel systems with conspecifics, tolerating them only in their vicinity when food is plentiful. These include desmans and the star-nosed mole, possibly also the American shrew mole. Generally, moles do not tend to make extensive migrations but remain within their established territories. There is little information about communication. Moles are generally not very vocal. Some squeaks are known, which are emitted by young animals or an irritation. Most underground living moles, like other burrowing animals, do not have a pronounced day-night rhythm. In the European mole and the Eastern American mole, the activity time is divided into several wake and sleep phases, with wake phases lasting about 4 to 5 hours each. Similar observations have been made in the star-nosed mole and Japanese shrew moles. Largely surface-dwelling moles can be both diurnal and nocturnal, although nocturnal activity often predominates, as is the case with desmans. Species in cooler regions do not hibernate. In winter months, the animals retreat to deeper soil layers. Moles are carnivorous and feed on a wide range of prey. The main component of the diet consists primarily of invertebrates, and in rare cases, vertebrates. Detailed studies are available only for a few species. Burrowing moles search for their food in near-surface tunnels. Their diet mainly consists of earthworms and is supplemented by insects and their larvae, centipedes, millipedes, and snails. Plants, mainly their underground parts, are added to a lesser extent. In surface-dwelling species, food is sought on the surface, partly in leaf litter. However, as with various golden moles, shallow tunnels may also be created in the soil. The diet spectrum is similar to that of burrowing representatives. Desmans and the star-nosed mole depend on bodies of water, where they swim and dive for prey. Insects like mayflies and caddisflies, as well as amphipods, form the primary source. Generally, moles digest their food relatively quickly. In the American shrew mole, the entire process from ingestion to excretion takes 45 minutes. Furthermore, their energy needs are relatively high, so the animals must consume a lot of food daily. This is especially true for species that dig underground. A 120g animal uses about 0.002 kilocalories for covering a distance of 1 m on the ground, while the same distance dug underground increases the requirement to almost 1.2 kilocalories. The daily food intake for the European mole and the Eastern American mole is almost equivalent to their body weight. The same may apply to golden moles, while in desmans, it is about half their body weight. Few species have internal energy stores, such as the star-nosed mole and the Japanese shrew mole, which can both store fat in their tails. For this reason, and due to the high food demand, the animals do not survive longer food breaks of 12 to 24 hours. Some species circumvent this by accumulating food reserves, such as the European mole. The body temperature of moles ranges from 34.5 to 38.4 degrees Celsius. The metabolic rate is comparable to that of other small mammals, but tends to be on the lower side in burrowing representatives. This is due to the high energy costs of digging and also protects the animals from overheating. Due to their dense fur, moles can only poorly dissipate excess heat. In warm environments, blood circulation increases, especially at the snout, tail, and feet. Burrowing animals then retreat to deeper depths, while semi-aquatic forms use water for cooling. Through their burrowing activity, underground moles as representatives of the fossorial, phodont, edifaun play a certain role in soil formation, pedogenesis. They contribute to the mixing of the humus-rich upper soil layer, a horizon, as part of bioturbation. Additionally, they transport humus-free subsoil material to the surface. Through their loosening effect on soil structure, they also promote aeration and, to some extent, drainage. Their burrowing activity also enhances plant growth and helps less competitive plants to thrive by opening up the vegetation cover. This, in turn, has an impact on the immediate animal world. Numerous other animals benefit from the tunnels created by moles. The semi-aquatic moles can serve as indicators of water quality and the integrity of the water network. The origin of moles is unclear. For a time, it was assumed they originated from the Eocene in Europe, which was then an isolated island. The oldest fossils are also known from there. They are assigned to the genus Talpidae, which is known from several tooth and jaw findings, as well as parts of the movement apparatus from the Isle of Wight and Hortle Cliff in the English county of Hampshire and from Creech Barrow in the county of Dorset. The findings come from the Heaton Hill Formation and the Bembridge Limestone Formation, both of which formed in the Upper Eocene. Talpidae, as the oldest representative of moles to date, shows certain characteristics that do not place it at the base of the family's development, such as specific dental features. The discovered foot bones also reveal certain specializations. For example, the metapodia and phalanges are shorter and flatter compared to shrew-like moles, considered the most primitive recent group. They indicate an early adaptation to digging properties, supported by the formation of a weak ridge on the upper joint end of the ulna, which is absent in shrew-like moles. However, the structure of the talus and calcaneus suggests more surface activities. Thus, moles already showed certain signs of a digging lifestyle in the Eocene, which likely consisted mainly of scratching and scraping the soil. This contradicts a partially held opinion of a semi-aquatic origin of the group. A significant difference from modern moles is that the tibia and fibula were not yet fused in Talpidae. The mix of primitive and developed features shifts the origin of moles to possibly the Paleocene and suggests an origin from present-day Asia. Moles' first evidence in North America dates back to the Upper Eocene. It involves a partial lower jaw with rear teeth discovered in the Florissant Formation in the Florissant Fossil Beds National Monument in the U.S. state of Colorado, assigned to the genus Talpidae. The genus name references the present mountainous character of the find region. The age of the deposits with the lower jaw is estimated to be around 34 million years old. Talpidae is still present in the lower Oligocene. Isolated teeth have been found in the Borgloon Formation near Butursum in Belgium. At the same time, moles can be traced in Asia. The Sanda Gol Formation in the valley of the Gobi Deserts in central Mongolia yielded upper and lower jaw fragments of Mongolopoda. The early Asian form is relatively similar in size and dental characteristics to the North American Talpidae. The findings indicate that moles already undertook continental-wide migrations during the transition from the Eocene to the Oligocene, reaching their current distribution areas. While the earliest moles cannot be assigned to any specific lineage, Persimigale represents a member of the Japanese shrew moles. The genus appeared in the lower Oligocene and is documented, among other things, with some teeth and toe bones from the Boulder Formation in southern England. However, the most significant discovery site includes several karst fissures in southwestern Germany, such as in Mohren northeast of Ulm, with some tooth and jaw findings from the Mohren 20 fissure indicating a presence as early as the uppermost Eocene. The form was geographically quite widespread and can be traced throughout the Oligocene in Central and Western Europe. Closely related to it is Myxomygale, which appeared slightly later but has a similar spatial occurrence. Both forms are characterized by a slender humerus. In Persimygale, however, the rostrum was significantly longer, which is often associated with minimal adaptation to burrowing activities. In general, the numerous karst fissures in southwestern Germany form a rich source for the development of early moles. The ones at Ehrenstein near Ulm yielded, among other things, dental and leg remains of Tegularoscaptor, a representative that is also closely related to the Japanese shrew moles. The rather slender humerus already shows certain characteristics, such as the broad elbow joint, which indicate a partially burrowing lifestyle. The Ehrenstein karst fissures additionally provided Geotropis, with which true moles are documented for the first time in the late Lower and Upper Oligocene. The animals were evidently already fully adapted to burrowing underground. Evidence for this is given by several partial skeletons, one of which comes from Inspel in Rhineland-Palatinate. The individual found there had the characteristic pre-thumb to widen the palm, and the forelimbs were relatively massive. In the Upper Oligocene, shrew moles can be traced, such as the genus Demonella from a karst fissure near Egingen, close to Ulm. The same applies to New World moles with Eugenia from the same locality. Both genera are, however, largely known only from teeth. Outside the southwestern German area, Desmans have their earliest evidence with Mygotalpa, with individual lower jaw fragments from the Upper Oligocene of northern France. Beyond Europe, further representatives of moles can be traced in the Upper Oligocene. In Eastern Asia, Mongoloscaptor is documented over individual lower jaw fragments from the upper sections of the Sanda-Gol Formation, likely in a closer relationship to the long-tailed mole. In North America, a lower jaw fragment from the Brule Formation in southwestern North Dakota represents the first evidence of shrew moles. The find is attributed to the genus Mystipterus. In the lower Miocene, moles diversified further. In addition to the still existing lineages, the family also produced the Suleimanini. Their characteristic form Suleimania is first detectable in Western Asia. Unlike all other moles, the hindmost upper molar was reduced and the corresponding lower one smaller. Superficially, the animals resemble the Dymelidae. Numerous tooth finds have been discovered at sites such as Harami and Gokler in Anatolia, with some fossils also found on the Balkan Peninsula. A previously puzzling form appeared with Niraga in the lower Miocene on present-day Sardinia. Named after the conical cusps on the lower molars, which somewhat resemble the Nurag fortress towers of Sardinia's Bronze Age. Desmonella remained a dominant form of shrew moles, persisting until the Upper Miocene and inhabiting large parts of Europe and Western Asia. In Western Asia, Desmonella shared the habitat with Theratiscos, which had relatively slender limbs like its relative. The genus is evidenced up to Central Asia. Within New World moles, Perscapenus replaced the older Eugenia and became an important representative. It is detectable until the late Middle Miocene in Western and Central Europe. Finds come from several karst fissures in southwestern Germany, such as Petersbuch near Eichstätt. Of great importance is also a partial skeleton from the lower Miocene deposits of Vusen near Regensburg. The massive humerus and the pre-thumb of the hand indicate the burrowing ability of the animals. Petersbuch also yielded remains of Leptoscaptor, which appears locally and sometimes in large numbers. In parallel with Myxomygale, Paratalpa, as a member of the Japanese shrew moles, is still evidenced in the lower Miocene. The genus soon disappeared again and was replaced by Desmanodon. The form is found in numerous European and Western Asian sites. Its slightly more robust humerus, compared to Paratalpa, suggests some digging abilities. This does not apply to the related Tenuabrachiatum, which appears locally in Petersbuch in the Middle Miocene. Another regional variant of the Japanese shrew moles is Pseudoparietalpa, reported from the Aral Formation in Kazakhstan. Desmans are represented in the lower Miocene by Megalia. The deposits from Vusan provided one of the few known partial skeletons, while individual teeth and jaw fragments were found in the karst fissures of Petersbuch. In the Middle Miocene, Asthenoscaptor and Gerhardstorchia also appear, the latter genus still existing until the Upper Miocene. Both forms are, however, rather rare. That's it for now. In the next episode, we'll take a closer look at Persian and the relationship with ninetails. Feel free to subscribe to our channel, leave a like, and check in next time.